Hi, everyone. My screen share is still working. You can hear me? Okay, perfect. Hi, everyone. I'm Elvisha. Thank you so much for tuning in today, and thanks for organizing this great initiative. So today I'm going to be talking about my work in machine learning and neuroimaging to better understand brain behavior relationships in healthy and clinical populations. White matter fibers in the brain directly and indirectly connect different brain regions. And these structural connections facilitate the flow of neural activation signals between different brain regions. These structural and functional properties of our brain underlie our unique and individual behaviors. Variations in our brain structure, activation, and connectivity are associated with individual differences in psychiatric and neurological diagnoses, dimensional symptoms, cognitive abilities, and personality traits. Traditionally, these relationships have been studied at a group level to identify differences between populations, such as healthy and patient groups, males and females, or children and adults. These analyses have led to monumental advances in our understanding of the structural and functional correlates of illnesses and behaviors. But with increased access to computational resources and large publicly available data sets, we can now implement data-driven machine learning predictive models to better understand these relationships at an individual level. In doing so, we can better predict illness risk, disease progression, and response to treatment for each individual. And that's exactly what I'm studying. But before we dive too deep into the details and the actual studies, I'm going to take a step back and provide a little bit of an overview and an introduction on machine learning and neuroimaging. So machine learning is the use and development of algorithms and models that are able to learn and adapt without following explicit instructions by analyzing and drawing inferences from patterns in data. In the context of neuroscience and psychiatry, machine learning approaches can facilitate predictions across binary, categorical, and continuous variables, and it provides a framework that we can use to understand the neurobiological correlates of behavior at an individual level. These algorithms can be characterized into three broad groups: descriptive, predictive, and prescriptive, and each of these have a different purpose in neuroscience and psychiatry research. The first kind, descriptive analyses, rely on observations to identify underlying patterns that can describe and summarize the data in an easily interpretable manner. These approaches are technically what we like to call postdictive, in that they retrospectively are detecting trends in existing data rather than prospective or future predictions. And these algorithms can reveal broad trends in the neurobiological, behavioral, and clinical phenotypes, and they've been used to identify overlapping and shared correlates of illnesses and behaviors. These analyses tend to be what we call unsupervised, meaning that they're not relying on existing labels of the data, but rather they're just trying to find patterns and trends in the data. So in the example of the visual that I have up, we're trying to figure out how that person in purple is related to the people in either the blue, green, orange, or pink. And we're not using individual information or the labels about the people, but rather we're just trying to see how they group together or cluster together and what are the patterns that are shared or unique between them. On the other hand, predictive analyses establish associations between independent and dependent variables in the existing data, and they use those associations to predict trends and properties in unobserved samples. So these predictive models have the potential to identify future risk for the development or deterioration of illness in a personalized manner. Examples of how these have been applied include those used to predict clinical, cognitive, and personality traits across distinct healthy and clinical populations. And unlike descriptive analyses, these ones tend to be supervised, meaning that they're relying on pre-observed ground truth labels that the model is using to learn associations with the input data before seeing how it can generalize to unseen data or samples. So in the visual that I have up, we now will have information about a group of people and their diagnoses, dimensions, cognition, or personality. We'll use information about those individuals and their behaviors to establish those associations. And then we'll have a model that we can then apply to this new unseen individual to predict their behaviors. Finally, we also have prescriptive analyses, which enable us to recognize different risks and offer potential treatment solutions. These approaches identify anomalies and suggest personalized interventions that can most effectively alleviate individual-specific patterns of concerns. Prescriptive analyses have been applied to predict clinical responses to medications and cognitive behavior therapy across psychiatric populations. Similar to the predictive analyses, these are also supervised because they first need access to labeled data, which tells them how different individuals and groups have responded to different solutions or treatments in the past, so they can now try and understand how new samples might respond to them. So together, we can use descriptive, predictive, and prescriptive analyses to get clinically meaningful insights that can then facilitate the development of personalized diagnostic tools and therapeutic interventions. One other thing we need to consider when thinking about machine learning is the types of algorithms or models that we're using. Classical machine learning algorithms are relatively simple linear models or nonlinear models that can be sparse or non-sparse in nature, while deep learning algorithms rely on one or more neural networks, which progressively extract higher level features from the input data to make predictions about the output. Although deep learning models can theoretically capture more complex relationships in the underlying data, simpler models tend to be more interpretable. And in fact, this trade-off between complexity and transparency or interpretability has been well studied. As model complexity increases, model transparency or interpretability decreases. Another way to think about this is as models become more complex, the bias that the models will have will decrease, but their variance increases. And this is a phenomenon known as the bias variance trade-off. Simpler models tend to be high in bias, which is often explained by underfitting of the relationship, while more complex models have high variance, which is attributed to their overfitting. And this bias variance trade-off is also closely tied to the balance between model transparency, which is the ability to understand the brain behavior relationships captured by the model, and the model complexity, which is the capacity to generate sophisticated predictions. Thus far in my research, I've mostly been focusing on predictive analyses using linear algorithms and neuroimaging, so that's what I'll be talking about today. And specifically, I use neuroimaging data in my predictive analyses, which means that I'm using information about the brain to then predict individual differences in diagnosis, cognition, personality, and symptom dimensions. So now let's shift gears a little bit and talk about the neuroimaging side of it. Neuroimaging is used to directly or indirectly image the structure or function of the brain. There are a lot of different techniques out there that are used for in vivo brain imaging and specifically in vivo human brain imaging. And some of these are shown here, where they're organized along the x-axis based on their spatial resolution and on the y-axis based on their temporal resolution. So certain techniques are more catered towards local or regional brain imaging, whereas others are designed for whole brain imaging. They also vary in terms of the time scale and the temporal resolution, which they can actually capture the data from the brain. So now I'm going to talk a little bit about the specific techniques that I use. And the first one is structural MRI, which qualitatively and quantitatively describes the shape, size, and integrity of gray and white matter structures. And it can be used to quantify properties such as total brain volume, surface area, gray matter volume, and cortical thickness. Another technique is diffusion MRI, which maps white matter pathways in the brain by measuring the diffusion of water molecules. Diffusion of water molecules in the brain is constrained by obstacles such as macromolecules, fibers, and membranes. And so that water molecules tend to diffuse parallel to white matter tracks or the structural tracks. So by measuring how the water is diffusing, we can figure out where and how big the white matter tracks are that are connecting different regions of the brain. Structural connectivity is something that we can derive from the diffusion MRI data, and it represents how connected two brain regions are through these white matter or neural fiber pathways. In other words, structural connectivity measures the actual structural or white matter connections between brain regions. Another MRI technique that we use is called functional MRI, which measures brain activity by detecting changes in blood flow that are coupled with neuronal activation. The brain is always rapidly consuming energy, but it does not have its own reservoir of energy. Instead, it relies on neurovascular coupling and the hemodynamic response for oxygenated blood to rapidly be delivered to brain tissue. And this functional MRI technique is measuring that ratio of oxygenated and deoxygenated blood, so you can indirectly measure how active a brain region is. Using this information, we can extract what's called functional connectivity, and this represents the correlation patterns between regional activation in the functional MRI time series. So in other words, using the local or regional neuronal activation patterns, we can look at different regions and correlate their activity to measure what's called functional connectivity. So functional connections are representing statistical correlations in the functional activity patterns. So that brings me to my current work, where I use structural, functional, and diffusion imaging data, along with linear machine learning algorithms to study brain behavior relationships between the brain and different behaviors, including cognition, personality, psychiatric diagnoses, and dimensions. And the main goal behind this work is to establish the functional and structural correlates of behavior across distinct populations. That way, we can better understand how potential changes in brain structure or function due to factors such as age, trauma, and injury can lead to behavioral disruptions. We can then also use this information to develop potential diagnostic tools and targeted or personalized therapeutic interventions in response to different illnesses. But there's also another layer to my work that I haven't quite touched on yet. So regardless of what specific brain behavior relationship we're trying to model, there are many different methodological choices that must be considered that can directly and indirectly affect the model's performance and interpretations. Some of these choices include the neuroimaging modality and the state that we choose to base our predictions on, the parcellations that we use to segment the brain into different regions, how we actually transform our brain imaging data, how many subjects we include in our sample, whether we use population-specific or general models, and I'll be talking a little bit more about that in a bit, the algorithms that we use, so are we using linear, nonlinear, or deep learning algorithms, and finally, the specific phenotypes that we're looking to predict. So in my work, I'm also trying to figure out how some of these choices can affect how accurately and interpretively we can model different brain behavior relationships. So with that being said, I'm going to present results from 3 different studies here, which have helped us understand how different structural and functional properties of the brain are related to cognition and behavior. So in the first study, we looked at how functional connectivity, which previously we defined as the measures of correlations of regional activation, and structural connectivity, so the actual white matter connections, are related to cognition. We also wanted to see if integrating information about the functional and structural connections into the brain into what we call a hybrid connectivity can strengthen that underlying brain behavior relationship, making it easier to predict cognition. And we did all of these analyses in a data set known as the Human Connectome Project, and it included 415 unrelated healthy young adults with 213 males and 203 female subjects. For all of these subjects, we also had their cognitive measures, which were measured using the NIH Toolbox Cognition Battery Task. In terms of the cognition, we were looking at measures of fluid, crystallized, and total cognition. Crystallized cognition is the ability to use learned knowledge, experience, and skills, and includes different aspects of language abilities. On the other hand, fluid intelligence is the ability to solve new problems using logic, encode new episodic memories, and adapt to novel situations in everyday life. And this includes measures of executive function, episodic memory, working memory, and processing speed. and total cognition is a summary of both fluid and crystallized cognition. And in order to model these relationships, we use linear ridge regression algorithms with nested cross-validation to actually optimize and understand what those relationships were, and then generalize those models to unseen populations. So getting into our results, here I'm showing along the x-axis the different cognitive scores we were trying to predict, and on the y-axis we have the amount of explained variance. So the amount of variance that the neuroimaging data was able to explain in crystallized, fluid, or total cognition. And in purple, we have the results for functional connectivity. In blue, it's for structural connectivity. And in green, we have hybrid connectivity. And the filled-in violin plots indicate that the models performed better than chance, and the ones that are not filled in or are white indicate that the models did not perform better than chance. So overall, we found that functional and hybrid connectivity are always able to predict cognition better than chance, whereas that's not the case with structural connectivity. We also saw that functional connectivity is generally able to better predict cognition compared to structural connectivity, and integrating the two into the hybrid connectivity does not increase prediction accuracy beyond what we already see with functional connectivity. So essentially, functional connections are more predictive of cognition than structural connections. We also then looked at the connections that were important and how they were related to one another. So here I'm showing a couple different heat maps. The first one that we have is looking at the different feature weights extracted for models using functional connectivity, FC, or structural connectivity, SC, to predict crystallized, fluid, and total. And on the on the left side, you have results from two different parcellations or two different ways to segment the brain. So on the top, we see that when you're using functional connectivity to predict cognition, it's generally pretty strongly correlated across the cognitive domains. The same is also true for structural connectivity. But when you're looking across functional and structural, the connections are not correlated with one another, suggesting that unique functional and structural connections are predicting cognition. The same is also true in the figure on the bottom of the left side, which was same analysis, but using a different set of brain parcellations. Again, functional connections are shared across domains, structural connections are shared across domains, but functional and structural are unique from one another. Then on the right side, we're showing how the network level connections are shared across the different models. So when we parcellate the brain, we tend to parcellate it into different regions, but groups of regions form what we call networks. So this may be, for example, the visual network. So these are regions that are responsible for the visual system or visual processing. And using the network level connections, we again see that functional and structural are unique from one another, but across the parcellations, so the top half and the bottom half, they are shared. So this tells us that regardless of the brain parcellation that we use, the same networks are going to predict cognition, which makes sense. But regardless again of the parcellation that we use, functional and structural connections will always uniquely predict cognition. So this now leads us to what are those actual functional and structural connections that predict cognition. So here, what I'm showing on the top are the functional connections that are positively and negatively associated with cognition. And on the bottom, it's the same thing, but for the structural connectivity. And again, we can see that the top and the bottom, those figures look very different from one another. And around the circle, you have different networks of the brain, and the connections between them indicate how strongly those connections between those networks contributed to the prediction. So for functional connectivity, stronger functional connections within the visual, somatomotor, and cerebellar networks, as well as between the visual and dorsal attention networks, predict lower cognitive abilities. On the other hand, stronger functional connections between visual, somatomotor, dorsal attention, ventral attention, frontoparietal, and cerebellar networks, as well as within dorsal attention and frontoparietal networks, predict higher cognition. If we're looking at structural connectivity, stronger connections within visual, frontoparietal, and cerebellar networks predict lower cognition, whereas stronger connections between limbic and subcortical and dorsal attention and frontoparietal predict higher cognition. And overall, although the details of the actual networks are not important, we can very clearly see that it is different functional and structural connections that are associated with cognition. And these results from this study support some of the other work that has looked at these relationships in a different way more recently. So in more recent work led by some of my collaborators, we've replicated the findings from the previous study. And specifically, we were looking at predictions of cognition, aspects of personality, and emotional traits based not just on functional and structural connectivity, but also on anatomical features. So this includes properties such as surface area, gray matter volume, and cortical thickness, and also tract-based features. So information about the integrity of the white matter structural tracts in the brain. And here, we've again shown that functional connectivity is more predictive or more accurately predictive of the different behaviors than structural connectivity or anatomical or tract-based measures, but they are all able to successfully explain some of the variance in our unique behaviors. And these analyses were done across two different data sets. So the first one is HCP, which is adults, and the second one is ABCD, which is children. So again, regardless of the brain measure you're using, they are to some extent able to predict different aspects of our behavior. But functional connectivity is always the most predictive one out of all of them. In terms of the connections that are important, our work also replicated the findings of this study, which looked at how cortical and subcortical structural and functional connections are related to a range of cognitive functions. So on the left side, they're showing the different functional connections that are associated with cognition, and on the right are the structural connections. And they found that there is a very small number of distributed structural connections, shown on the right, and a larger set of cortico-cortical and cortico-subcortical functional connections that express the association. And again, the functional and structural connections are very distinct from one another in terms of how they're related to cognition. So to summarize so far, we've seen that functional connectivity is more predictive of cognition than structural connectivity, and integrating the two does not improve predictions. We've seen that distinct functional and structural connections are able to predict individual cognitive abilities. So altogether, these results suggest that studying both function and structure is important because the two are uniquely related to cognition. And doing so provides a more comprehensive insight into the actual brain bases of cognition. But we don't know if these relationships are consistent across populations. And specifically, I'm interested in considering whether these relationships are shared or unique across males and females. So some of the work that's already been done out there suggests that males and females are differentially predictable in terms of their IQ. This study looked at whether cognitive abilities were predictable in males and females based on functional connectivity, and they found that female IQ might actually be more predictable than male IQ. And specifically, they're showing that the correlation between true and predicted values is higher in females than it is in males. The same study also looked at the unique associations between connectivity and cognition in males and females. So on the left, you have the connections that were important in females, in the middle is males, and on the right is a summary. So the ones in red are the ones that were important in females, the ones in blue in males, and the ones in black are the ones that were shared. And you can see that there are only 3 connections that were shared and equally important between males and females. So this study concluded that males and females actually have unique associations between connectivity and cognition. And I wanted to see if that was actually true and if we could actually replicate those findings. So in this next study, we looked at sex-independent and sex-specific relationships between functional connectivity and cognition in 392 healthy young adults. And these were all male-female pairs. And for each subject, we had their functional connectivity, and we also had their cognitive composite scores, so the same ones I mentioned before, as well as their individual task scores. So these individual task scores represent things like cognitive flexibility, inhibitory control and attention, episodic memory, vocabulary, reading, working memory, and processing speed. So instead of looking at just the broader summary cognition scores, we also wanted to look at an individual task score if those were similar between males and females. And once again, we use linear ridge regression models, and here we were looking at sex-independent and sex-specific predictions. In other words, we trained models that were either had access to the entire data set, so males and females, or they specifically used information about males or specifically used information about females, so that way we could capture general relationships, but also sex-specific ones. And once optimized and trained, these models were tested on both sexes, so we could also see if the models generalized across sexes. So diving into our results, these are our results from sex-independent models, so models that were trained on both males and females. Along the x-axis, we have the different cognitive scores that we were predicting, and on the y-axis, we have the prediction accuracy, which is the correlation between the true and predicted scores. And shown in blue are the results when the models were tested in males and in pink for females. And once again, the colored in violin plots indicate that they perform better than chance, and the ones that are not colored in indicate that they were worse than chance. So looking at the extent to which sex-independent models were able to predict these scores, we found that the total composite cognition score is equally predictable in males and females, as are the crystallized composite and crystallized task scores. But fluid abilities are only accurately predicted in males, if at all, and not in females. So given that we have meaningful predictions for both total and crystallized domains, suggests that those models are capturing some sort of shared brain behavior relationship. Whereas for the fluid domain, the inability to predict fluid scores in females suggests that the model might actually be capturing male-specific brain behavior relationships. To dive a little bit deeper into that, we can then look at the results from the sex-specific models. So again, we have the cognitive scores along the x-axis, prediction accuracy on the y-axis, and here we have models that were trained and tested on males in purple, trained on males tested in females in blue, trained on females tested in males in green, and trained and tested on females in orange. And again, we see that the predictions in the total for the total composite are equally comparable and successful in both males and females, regardless of who the model is trained on. The same is also generally true for the crystallized abilities, but the model generally fails to predict fluid abilities altogether. So here again, we see that the meaningful predictions in both sexes for total and crystallized abilities suggests that both male and female specific models are capturing shared or overlapping brain behavior relationships. But for fluid abilities, it seems they're struggling to capture any meaningful relationship altogether. So then we can actually look at what those connections were. So here we're showing on the left the connections that were positively and negatively associated with crystallized and fluid cognition for females, and on the right is for males. So again, around each of those individual circles, we have the different networks, and the edges between them tell us how strongly those network connections contributed to the predictions. So we see that functional connections between visual, dorsal attention, ventral attention, and temporal parietal networks are associated with better crystallized and fluid abilities. We also have that functional connections within visual, somatomotor, and temporal parietal networks are associated with weaker crystallized and fluid abilities. But if we look across the figures for males and females, we generally see that they look about the same. And although there are slight differences in the strengths of these associations between the sexes, so in some cases, those connections are more or slightly more or less important in the models for males and females, the general trend observed is consistent, suggesting that brain behavior relationships between functional connectivity and cognition are generally conserved across the sexes, at least in the specific population that we were studying, which was adults. So now, to summarize what we have so far, we've established that functional connections are more predictive of cognition than structural ones. We've seen that functional and structural connections that predict cognition are unique, but the functional connections that predict cognition are shared across males and females. So this now leads me to my next question of what about age groups? Well, once you start thinking about age, you also need to consider that our brain, as well as our behavior, is not the same throughout the lifespan. So for the next study, we decided to focus on the structural properties of the brain instead of the structural and functional connections, since the brain structure is what is most consistently, or is what is most strongly changing throughout the lifespan. So for a little bit of background, the brain is constantly changing in terms of volume, surface area, and cortical thickness throughout our lifespan. Cortical thickness, which is shown in the purple dashed line, rapidly increases before birth, peaks in early childhood, and then gradually decreases throughout the lifespan. Total cerebral volume, or total brain volume, increases quite a bit until early adolescence and then begins to slowly decline. Finally, surface area of the brain also increases until adolescence before slowly declining. So this suggests that if there are underlying relationships between anatomical properties and cognition, which several papers have shown that they do exist, these relationships must be changing in line with these trajectories or somehow accounting for these trajectories in the neuroanatomical property changes. We also need to recognize that these changes in neuroanatomy differ between males and females. Males tend to have larger volumes than females in terms of gray matter, white matter, subcortical gray matter, and ventricular volume. And these trajectories throughout the lifespan also show that those differences between males and females are generally consistent throughout the lifespan. So this now suggests that any existing relationships between anatomy and behavior might also be sex-specific. So in this next study, we relied on two large-scale data sets. First, from the Human Connectome Project, we had 1,013 young adults. And then from the Adolescent Brain Cognitive Development data set, we had 1,823 children and adolescents. And for all subjects, we again had their composite cognition scores, so total, crystallized, and fluid. And this time we also used, again, the individual task scores. From the structural scans of these participants, or the structural MRI, we extracted their surface area, gray matter volume, and cortical thickness across 400 different brain regions. And again, these different brain regions can also be assigned to the networks to which they belong. And just to clarify, surface area represents the actual surface that a given region occupies on the brain surface. Gray matter volume is a measure of the total volume that is occupied by that region. And finally, cortical thickness measures the average thickness of a region in the brain. Once again, we were using linear ridge regression models to predict the composite and task scores. And this time again, we were using surface area, gray matter volume, and cortical thickness for those predictions. Here, we trained sex-independent and sex-specific models for each data set. By that, I mean that for each data set, we had one model that included all of the participants, and then we had a male-specific one and a female-specific one. And once optimized, the models were evaluated across both data sets and both sexes. So getting into our results, this is now looking at how the models performed in HCP, or the adult data set. And here we have accuracy shown along the x-axis, and the different cognitive measures are ordered along the y-axis. And we have the results for surface area, gray matter volume, and cortical thickness. And overall, first of all, we wanted to see if those measures could actually even predict cognition. And we showed that, yes, they can successfully predict cognition. As part of the study, we were also doing an additional set of analyses where we were looking at whether correcting for ICV, or intracranial volume, would bias these predictions. So I showed previously that our brain volume is constantly changing throughout the lifespan. So when we're considering different populations that have known differences in brain volume, such as males and females, or kids and adults, we need to see if we want to correct for those individual differences. So as part of the analyses, we were looking at how that correction would affect the results, but I'm not going to go too deep into that. Just another brief side note, for all of these analyses, we found that the results were pretty comparable if we use the sex-independent or the sex-specific models. So to make it clear, I'm going to just stick to the sex-independent results, but know that everything was replicated for the sex-specific ones too. So now going into the ABCD dataset, which is the children and adolescent dataset, we again see that surface area, gray matter volume, and cortical thickness can successfully predict cognition in children. And all of these models also seem to be able to generally generalize across the population, so from kids to adults or adults to kids. Where it got interesting was once we started looking at the actual networks that the model identified as being important for the predictions. So when we're looking at how surface area and gray matter volume can predict different cognitive scores, this is a plot of those feature weights. And along the x-axis, we have the different networks of the brain. And on the left side are networks that are typically thought of as heteromodal or sort of association networks that integrate information from a lot of different brain regions. On the right side, we have networks that are unimodal or somatosensory, which tend to be more specific to each modality. So for example, the visual network primarily focuses on visual processing. Somatomotor is very much focused on our somatosensory and our motor abilities, and so on. Whereas the heteromodal networks on the left side are going to integrate information from those unimodal networks. So here we're showing the feature weights in the HCP dataset or the adults, and we see that surface area of visual networks and gray matter volume of default mode, language, and control networks are associated with cognition in adults. In kids, we see that it's the surface area of default mode and somatomotor networks and the gray matter volume of visual, somatomotor, and dorsal attention networks. So both of these are pretty similar so far, but where it gets really interesting is when we started to look at the cortical thickness weights that were associated with cognition. So here we see that there are opposing gradients that emerge for adults and children for the associations between cortical thickness and cognition. So in adults, we see that heteromodal association networks, like the default language and control networks, are more important than the unimodal sensory ones. But in kids, we see the exact opposite. So the unimodal sensory ones, such as visual and somatomotor, are far more important to predict cognition than the default mode networks or language or control. And these specific brain behavior relationships that we do capture and the unique patterns that they exhibit in the two data sets are in line with some of the prior work that's shown that there are nonlinear maturation trajectories for cortical development. So essentially, when you're looking at how the brain is developing throughout childhood and adolescence, there are marked trajectories that have been established such that unimodal or somatomotor networks achieve maturity earlier, followed by the more association or heteromodal networks that mature much later in adolescence. So it suggests that those relationships are also present when looking at not just how they're developing on their own, but also how those networks are associated with our actual behaviors or our cognitive abilities. So to put it all together, these are our findings. Functional connections are more predictive of cognition than structural ones. The functional and structural connections that predict cognition are unique, but those functional connections that do predict cognition are shared across males and females. The morphological properties of our brain or the structural properties of our brain can also predict individual cognitive abilities. But once you get to those structural properties, you see that there are differences between different age groups. So adults and children have unique associations between cortical thickness and cognition. So with that, I hope I've been able to show you how you can actually use machine learning and neuroimaging to better understand the neural correlates of cognition. And I'd just like to end off by giving a quick thank you to all of my mentors. So Avram Holmes, the entire Holmes Lab, Thomas Yo and the Computational Brain Imaging Group, Amy Kusieski and the Computational Connectomics Lab, as well as all of my funding sources. And thank you for tuning in today.
All right, fantastic. That was a very informative talk. There's a lot of really cool work that you're doing. Let's jump straight into the questions here. So on slide 25, question is, what is considered a good amount of explained variance? Is it just that you need to enough that you're better than chance, or is there like a goal value that you're headed toward?
That is an excellent question. So a lot of it depends on what it is that you're looking for. For reference, traditionally, when we think of when genome-wide association studies or GWAS studies became a thing.
Mm-hmm.
A lot of times it was considered great if you could explain even like 1-2%, like 5% explain variance based on your genetics is a lot.
Okay.
We can sort of have a similar approach when we think about the brain, because it's important to recognize, obviously, that the brain is not, or at least the properties or connections in the brain are not necessarily the only thing that determine our behaviors.
Mm-hmm.
So these explained variance values that I'm showing are pretty comparable to most of what's been reported in literature as the high end. There is a lot of work, actually, if we look at this one. So these are showing correlation values instead of explained variance, but basically this translates to the fact that for cognition, you can explain as much as about 20% of the variance in behavior using the brain.
Mm-hmm.
But once you get to more perhaps complex behaviors, so things like personality, emotions, mental health, that number drops down quite a bit. So
okay.
If you get down to even like a few percent, 5%, that's considered really successful. And it's just a testament to the fact that there are a lot of other environmental factors or social factors that are going to strongly influence these behaviors, and perhaps more so than the brain aspect themselves.
Okay. So I guess to follow up on that question, so you mentioned all these additional factors beyond just the connections of the brain. Is there the potential, at least, for those factors to also be studied in a similar method to what you've shown here in this talk?
Absolutely. So more recently, there is a bigger push to sort of consider how those additional factors, so this could be things such as demographics, socioeconomics, education, different life experiences contribute.
Mm-hmm.
And some people specifically are looking into developing models that integrate that information on the modeling side as well, or models that are specifically based just on those factors that are let's say, brain related.
Okay.
I would strongly believe that depending on as much information as you can integrate,
Mm-hmm.
the more likely you are to be able to explain more variance.
That makes a lot of sense. All right. Let's see. So on slides 34 to 36, what can be done to improve the sex-specific models, if anything?
That's a great question. So right now, we are sort of limited in the analyses that we can do with the sex-specific models because we only have information about the self-reported biological sex. We don't have any information about gender. We don't have any information about hormonal status. We don't have any information about all of the other underlying factors that might be contributing to the sex differences. And another point that I'd like to mention is that in the same way that there's only so much variance we can explain in these behaviors based on brain data, a lot of the variance that we are saying is a sex difference, I think, is also very much going to be affected by social factors. So more so than necessarily the biological differences between males and females, we need to recognize that males and females are socialized very differently even before the time that they're born. So males are often like, okay, like we'll get everything in blue or like everything in pink for females. They're socialized very differently in school, whereas males are perhaps more encouraged to participate in sports. And females might be more like pushed towards the arts. So I think a lot of these differences also need to account for the fact that there are probably cultural differences in how males and females are socialized.
Mm-hmm.
And if we consider all of those factors, I think these models would be far more accurate.
Okay, that's interesting. Let's see, on slide 37, can you talk a little bit more about how connections between the different systems are established?
Yes. So in terms of the functional connections, which is what we were looking at here, I can actually go back to this slide here.
Mm-hmm.
So the way that these functional connections are first measured, like I mentioned, is we're looking at how similar different regions are throughout the brain in terms of their activity pattern. Okay. So one thing that is sort of a traditional saying in neuroscience is neurons that fire together wire together. Okay. So essentially, if two neurons have similar functions, they're going to be more correlated if they're active at the same time. they're going to fire together more often. So essentially, these functional connections are established through learned experiences. So as different brain regions realize, okay, when that one's doing something, usually I have to do something too. So these connections become stronger in that sense.
Okay, that makes a lot of sense. And the last question here, a little bit kind of open-ended, but I think it's an interesting one.
Yeah.
Why are the fluid tests so much more difficult to predict accurately than the
That is
crystallized ones?
Yeah, that's a great question. So, our hypothesis for that, which there's some literature out there to support it, is that if you look back at this slide, I mentioned that crystallized cognition is the ability to use learned knowledge, experience, and skills, whereas fluid cognition is the ability to solve new problems. And it's more about flexible thinking and like in the moment thinking.
Okay.
So you can't necessarily train yourself to have a much better processing speed or working memory or executive function in the same way that you can very much improve your language, you can very much improve your vocabulary, and so on. So we think that because fluid abilities are perhaps more variable and more state dependent, so perhaps if you got a little bit more sleep one day or have had more caffeine in a given day, or just like a series of different factors that aren't necessarily just entirely in your brain, but also external factors, then those are more likely to affect your fluid abilities. Whereas if you know what a word means, it's probably not going to change whether or not you know what that word is, depending on how caffeinated you are or how much you've slept. So we think that because crystallized abilities are more shaped by learned experience and knowledge that you develop throughout your lifespan, those same experiences are also shaping your brain connections. So there might be some sort of convergent change that's happening, whereas as you're learning new skills, your brain connections are also adapting in response. For fluid abilities, because it's more sort of like state dependent, it might just be more variable, and thus it's harder to establish those connections.
Okay, that's interesting. But I mean, but at the same time, it makes a lot of sense that it'd be more difficult to do that.
Yeah, I mean, someone might be working on something that completely disproves that, but that's just our hypothesis on that for now.
I mean, that's a fair hypothesis. So I think it's an interesting field, and it'll be really cool to see, as things continue to progress, how it sort of shapes up.
Definitely.
I think that's all the questions we have. So thank you again, Elvisha, for your presentation. Like I said, just really super interesting stuff that you're doing. And
Of course. Thank you so much for having me. And thanks everyone for tuning in.
absolutely, thank you again. And I'll get you to hang around for just 1 second. We'll talk very briefly. For
Sounds good.
everybody else, we will be back at about 1 o'clock with our next presentation.